Okay, so we are live for group
The
2.
last time.
The last one. Okay.
All right.
Problem number 1. Okay. If you look at problem number 1, what is it asking? It's asking you to draw the gel and then kind of just asking you different questions. So the gel kind of looks like this. That's where you would see the G's, the C, A, T, and C line up according to where they are in their in the lineup of things. Question B is asking you what happens if you use E. coli DNA polymerase 1 during the sequencing reaction instead of Taq polymerase. Well, Taq polymerase is used in PCR experiments because it can withstand heat. So if you were to use the E. coli DNA polymerase 1, it would be denatured, and then you wouldn't have a functioning DNA polymerase. What happens if you have E. coli DNA helicase? If you add that into the normal sequencing reaction, we said it would just kind of be an extra DNA helicase. It could potentially break down as well, and it wouldn't cause any problems. It would have no effect. If you forgot to add the DDGTP, DDTTP, and DDCTP, and only added DDATP, you would only be able to see the ATP show up. The A's on, you would only see them in the column. You wouldn't be able to see any of the other G, T, or C's. And so you would have an inaccurate gel electrophoresis. Question 2 is just a fill in the blanks. So a typical PCR cycle, the third step is done at 72 degrees, while the second step is done at 52 degrees Celsius. AMPR is a common selectable marker that is found in many plasmid vectors, while restriction endonuclease recognize and cleave DNA molecules at palindromic sites. During agarose gel electrophoresis, DNA is placed in an electric field. DNA migrates towards the positive electrode during the electrophoresis. DDATP lacks the 2' hydroxyl group and the 3' hydroxyl group. Transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. During blue-white screening, IPTG is commonly used to induce the expression of the lac operon, while XGAL is cleaved by beta-galactosidase to turn E. coli cells blue.
Sweet. Number 3 is beginning to be a really cool problem. So this is one of the applications, applying our genetic knowledge to do awesome things. And more specifically, that awesome thing is helping to perpetuate a PCR reaction or polymerase chain reaction. So actually, in my research this semester, we had to come up with 2 primers to help synthesize 2 genes that we extracted from zebrafish DNA. So I got to do this for reals, and it's really cool. And who would have thunk that at BYU Idaho we have all these really cool high-tech toys? But we do, so it's really fun. In number 3, 3A, it's asking us to identify the sequences that you would use for the 2 primers used in this PCR experiment. So you have to look and pay attention to the 3' ends of both strands. So here, hold on, let me share the screen with that stuff on it. Here it is. Okay. So we're looking at the 3' ends right here, not the whole thing, just here, and then just here. So we're looking at those bold bases there, and from the first one we see, where is it? Here it is. So for this section here, we do the complementary strand for that primer, and that it's going to be going obviously from the 5' end to the 3' end because it's anti-parallel there, and you get the GAGCCTTCACTCAGTC primer for that bottom strand. That's the primer for that bottom strand. The second strand, the one on top, we're looking at the 3' end there, and we're looking 5' to 3' here, and 5' to 3', and that top sequence, that sequence for the second primer is going to be TCGGAGTTAACGTGAC, 5' to 3', the complementary strand of that area for the primer to go. And that's how you do that part. Pretty simple, which is really awesome because all this crazy stuff of cloning and amplifying genomes is, we all thought it was really difficult, but in all reality, it starts with these little basic principles, and that's pretty mind-blowing. But in order for this to happen, you have to have the right annealing temperature, or your primers won't stick to your sequence, and all of a sudden you can't carry out that PCR reaction. So it's important to understand what temperature should we run our thermocycler at happens correctly. So B is asking that same question. Well, what are these temperatures we need to run these primers at? And so it gives us the equation here to determine the temperature in Celsius right here. So we see that the equation is, it's expressed as 81.5 plus the quantity of 0.41 multiplied by the GC content, talk about that in a second, minus the quantity of 675 over the number of nucleotides within that primer. Okay, so N is, just as we stated, is the number of nucleotides within the primer. The GC content here is that percentage of GC nucleotides in the primer expressed as an integer, as in a percentage, not as the decimal, before you multiply by 100. So let me share the other screen with you. Other screen. Really quick. Here. Shift. All right. So right here, number 3. Oop, everything blue. So we did just that. So primer 1, we counted all the G's and C's and all the nucleotides together, and we have 9 over 16. That yields 56%. We stick that in for where our GC content is, and we stick in 16 nucleotides total where we stick the N in. We plug and chug, and voila, you get the temperature in Celsius of 62.3 degrees Celsius. We do the exact same thing on the bottom, and instead we get 8 over 16, so that content, that GC content's of 50%. So we plug and chug just like we did before, and voila, you get the second temperature as 59.81 degrees Celsius.
Nice. So problem 4, do I have the picture of it up? Yeah, okay. So you would draw the gel electrophoresis, and that first lane is just showing, it's kind of showing from 1, 2, 3, 4, 500, and then 1,000 base pairs. And then 1, 2, 3, 4, 5, and 6 match up with the restriction enzymes used. And so you can see that's just where you saw the cutting of the different numbers of base pairs. So at 1, this is 50. This is, what is it, 300 and 500, 350 and 500 right here where they were cut. And so that's just showing you on a band, like where they would sit. So B, she showed in a video this week, and it's just talking about where the restriction enzymes cut the DNA. And so you see the BAMH1 cuts it at 50, then 350 base pairs from that, and then there's 500 base pairs left over. And then you see down here and then you have that for EcoR1 and HinD3. And when you see the combination, you see BAMH1 cuts at 50, and again with 500 left over. And then the EcoR1 cuts that 350 into 2 different quarters, and same with HinD3. And so that's how you were able to get the map and see, because you know that the BAMHI comes back down with both of them, and that the HinD3 and the EcoR1 cut in between those 2 sections, if that makes sense, to see how many base pairs are in between the cuttings. Okay, and then 5.
5 is another really cool application problem. I just love this. I love doing the application part. It's really fun. I love it. So that's more incentive for Sister Mayo to grade leniently if I got this wrong. But okay, here we go. Step 5, question 5 is asking, let me just look at the question really quick. So it's asking, so we're studying our favorite gene, and that we found in some species of yeast. And so it's asking, how on earth would we clone a version of this gene into a plasmid vector? And then it puts on the kicker, and how can we tell that this recombinant plasmid vector would have our gene in there for sure? So that's what number 5 is asking. Well, what would we do to stick this piece of DNA into our plasma vector and then make sure it actually happened? So that first thing you got to do is you've got to isolate your favorite gene. So however you wish to do that, you can do that in a variety of different ways. You isolate your favorite gene, and in a lot of different scientific disciplines, they use kits. So it's not as it's very user friendly now. So you don't have to go through and use all these crazy organic solvents to extract the gene and its mRNA sequence. So what you first have to do here is, I'm showing here right here, is you have to convert your mRNA to cDNA, your chromosomal DNA. So you purify that mRNA, and then you have to mix that mRNA with a primer. So this is like preliminary PCR amplification. So we have to determine, well, we've got our favorite gene. This is its mRNA sequence. We've got that. It's pure. We know we're not messing with any other crazy miscellaneous bits and chunks of sequencing in there. So we want the pure stuff. We identify primers so that we can run a PCR reaction on it. You add your reverse transcriptase and your energy to transcribe those individual base pairs. You also add your rRNase to digest, the stuff that we didn't actually really need. So we're getting the sequence as we need it. The raw, not the raw uncut. We actually RNase cuts the raw. So we have the purify. This is what we want. Like, it's the spark notes of your DNA sequence. And then you add your DNA polymerase from E. coli, and you amplify that. And so you've got chromosomal DNA. So that second part is, we've got our chromosomal DNA now. So how do we get it stuck into the plasmid? So this is kind of where I left out the part for the blue-white screening, just to kind of get the the idea of what's going on here. So the idea is we've got to have our plasmid and the chromosomal DNA we've come up with. Now what we got to do is we got to digest those both with a restriction enzyme. Now what restriction enzyme are we using? So that's dependent on where we want to stick it, or where we want to put our favorite gene, and exactly in that plasmid. So the next step after that is you have to cleave. So once that cleaved plasmid and chromosomal DNA are mixed, that chromosomal DNA is inserted into the plasmid. And then, of course, you add your DNA ligase, sticks it all back together. After that, you have to introduce your recombinant plasmid into some competent E. coli cells. Competent E. coli cells meaning that they're going to be able to take that DNA, replicate, and make sure and to preserve that sequence within that DNA sequence. So, and then the last step is you got to grow it on a growth medium with ampicillin, and then from there you can determine if that recombinant plasmid was successfully inserted into that E. coli bacterium. Now, the reason why I left out, so with the blue-white screening here, so you have to enter your favorite gene into the mix of the chromosomal DNA and the plasmid and the restriction enzyme. So right here in number 2, in step 2, you have to stick in that favorite gene as well. It's a lot like what's the gene we stuck in there for? It's a lot like beta globin. You have to stick in the beta globin gene. In this case, it's your favorite gene, and with the whole mix, with the plasmid and the chromosomal DNA and your restriction enzyme. So here in the second step, in the second facet in the second step, you've got your favorite gene stuck into a vector that's been inserted with your chromosomal DNA into the plasmid. And then your DNA ligase kind of stitches it all back up together. And that's just the very simplistic style of what's going on when you're doing a cloning experiment, is when you stick that gene in, from what I understand. And just gotta throw it out there. I got this sweet book. It's called The Gene by Dr. Siddhartha Mukherjee. And if you've ever wanted to learn anything about cancer or anything, Ken Burns did a what's it called, a documentary on cancer, and he talks all about this. And in his book The Gene, he talks about the beginnings of how this started. And this really simple question here, number 5, is like the precursor of this giant thing called CRISPR, or CRISPR-Cas9. And if you need insulin injections, they used to have to go round up all the stray dogs and all the cows that you didn't, and all the cow pancreases that weren't consumed when they were killed for making your hamburgers. Overgeneralization. And they have to grind up all those pancreases and extract insulin from those pancreases from dogs and other animals. And so if you were a diabetic way back in the day, your insulin was like buku bucks. But after 1970 and into the 80s, around late 70s, 80s, they figured out what this thing called CRISPR-Cas9 is. Instead of having to use a virus, they found out that you can just stick these right into E. coli cells, and you can stick in that gene that encodes proteins that makes insulin into E. coli cells, and then you just take your insulin from your E. coli. And so if you need insulin, a lot of your insulin comes from genetic engineering. So I hope you don't have anything against eating genetically modified corn, because your insulin definitely is definitely genetically modified and synthetically produced. It's E. coli. So really cool. And that's the end.
Very nice.